Okay, now let's move on to transduction. So transduction is when DNA from one bacteria is transitioned to another bacteria via a bacteriophage infection. So what is a bacteriophage? A bacteriophage is a virus that infects bacterial cells. And so bacteriophage actually look kind of like really weird little robots. They have these little fibers, these tail fibers. They have a protein coat. Again, sorry, I have terrible handwriting. It's really hard to write with this pen. They have a protein coat, and inside they're going to contain genetic material. Now, that genetic material in bacteriophage oftentimes is going to be DNA, but like other viruses, that genetic material might sometimes be RNA. And so the idea is that when a bacteriophage infects a host cell, one of the genes from the bacteriophage actually causes the destruction or the fragmentation of the host DNA. That's not good, because what that means is that the host DNA cannot be expressed anymore. So the idea is you have a bacterial cell with its chromosome here, and after phage infection, that cell has basically all types of just chunks of DNA floating around in the cytoplasm. Eventually the cell lyses or explodes. So there are two different types of transduction that occur after phage infection. There is generalized transduction, and generalized transduction is just any bacterial DNA being transferred to another bacterial recipient cell. Okay, the recipient will be a living cell. The DNA comes from a cell that had been infected. Okay, so the cell gets infected, DNA gets sliced up, cell explodes, releasing the cellular debris, including the DNA, out into the environment. All right, that environment can be water, it can be under your skin. That environment can be anywhere that bacteria and phage exist. Now, specialized transduction, this is kind of a fun little subject, because this is where only certain genes get transferred to particular cells. Why does that happen? Well, that's a big question. Basically, what happens is that viral DNA becomes a part of the bacterial chromosome. All right, this happens a lot in a lot of viral infection period. In fact, in humans, there are viruses, say like the varicella virus. Varicella virus gives us chickenpox. So when you get varicella virus, you'll get chickenpox, all right? You get all the little sores all over your skin. But for some reason, the varicella virus can also attack cells in your spinal cord. The motor neurons in your spinal cord allow you to move your muscles, allow you to flex your fingers to write, allow you to move your arm to pick up, or allow me to move my arm to pick up this pen, all right? The varicella virus can become incorporated, or at least the viral DNA can become incorporated inside of the DNA of the host, the human host, in the cells in the spinal column. Later on in life, even as a teenager, 20s, or even later on in your 60s, if a stressful event happens, the varicella virus DNA can become expressed, and that results in shingles. And so shingles occurs in a patch of tissue, usually on only one side of the body, where the cellular infection where the cellular uptake of viral DNA had happened, and viral DNA integration into the host chromosome. Crazy, right? So I had chickenpox when I was a kid. I know that somewhere in my spinal cord there's a certain number, who knows how many, motor neurons that have the varicella virus DNA incorporated into my own DNA. Someday, if I'm unlucky and I've been under a lot of stress, I may develop shingles based on that early infection. This happens also in bacteria. When viral DNA integrates itself into the bacterial chromosome at a given site, the site is very specific for each integration, the viral DNA then hides in the bacteria. So, here's a picture of of special transduction. So the phage here infects, let me get a nice bright color for us to work with. the phage particle, the phage virus, infects the cell, injecting its DNA. And over here, they're showing that this particular bacterial DNA has genes to incorporate histidine and lysine. Then the expression of the viral DNA, which is the red stuff here, and the replication of the viral DNA causes the destruction of the host DNA. What usually happens in general transduction, okay, this is general, is that the cell then goes on to reproduce the virus itself. So all these viral DNA strands have the instructions to make new virus, these little protein coats, and to package new viral genomes inside the protein coats. The cells lyse or explode, releasing all the new viral phage out into the environment, but also releasing these little pieces of DNA. Interesting, right? Now, this phage every now and again can pick up a piece of the host DNA inside of its head. So instead of picking up, so what usually happens is you have a phage that's being constructed, all right? And normally you would have phage DNA being put in here. But every now and again, you have a mistake happen. When that mistake happens, instead of phage DNA, we end up with bacterial chromosomes, bacterial DNA being put inside the phage head. Now, the phage is still going to go around and infect like it normally does. So here's the second phage infection. But in this case, instead of infecting with phage DNA, it's injecting the DNA from the former bacterial cell. So now what happens is the new DNA from the old, let's say, bacterial 1, bacteria number 1 becomes incorporated into the genome of bacteria number 2. And now you have an expanded genome. How crazy is that, right? So this can happen with all kinds of critters. P22, phage 22, so phage is usually indicated with P. So phage 22 infects salmonella. P1, phage 1, infects E. coli. And the reason why it's called P1 is because it was the first phage discovered while working with E. coli. And guess what you can do with this? Map as well. So after transformation maps were discovered, people started figuring out, hey, we can do that also using these phages. So what they did in one experiment was they looked at the size of DNA that can be packaged by phages by looking at each phage and how much of an individual chromosome it can take with it. So P1 can take up to 2 to 2.5% of the entire E. coli chromosome. P22 can take 1% of Streptococcus chromosome. And so what ends up happening is this allows you to experimentally see how much chromosomes transferred from one cell to another, and you can map what genes exist where. And so if you look at 2 E. coli strains, a donor that's arginine, methionine, and streptomycin sensitive, okay, so this would be your prototrope again. Oh, let me get my pen. There's your prototrope. Here's your auxotrope, okay? Arginine negative, methionine negative, streptomycin resistant. In this experiment, they used P1 phage to infect a donor, the prototrope. They allowed it to sit there for a little while. They allowed lysis to happen. Then they took a new strain, the negative strain, the auxotrope, and they allowed the secondary phage. All right, so here's the first round, the regular phage infecting. The occasional phage will pick up a piece of bacterial chromosome, and then on the second round, you can map where the arginine and methionine was integrated. So here's some larger pictures showing you how that all works out. Co-transduction is another way to do this. Dr. Wu was the first to come up with the idea of co-transduction, and this is where you, in addition, so basically you're transducing one strain with another strain's DNA, and based on the amount of time that you allow phage infection to occur, let's say, 5 minutes, 10 minutes, and so forth, you can start mapping information off of chromosomes. You just need to know what that you can use transduction in mapping as well as conjugation. Now, for the last topic, we're going to be covering transformation. Transformation is a process by which a bacterial cell absorbs or takes up DNA from the environment, from dead bacteria, okay? So even though you kill bacteria on your hands using, let's say, hand sanitizer, which, by the way, isn't that effective, when you kill bacteria on your hands but you don't wash it off, you still have bacterial DNA on your hands. So any new bacteria that get on your hands can absorb that. Frederick Griffith in 1928 was the first to come up with this by researching a type of Streptococcus pneumoniae, which actually gives us pneumonia. Now, pneumonia is a condition. It's a disease, but pneumonia is actually the condition of having fluid in your lungs. Now, there are two types of transformation. There's natural transformation, where DNA is taken up from the environment, and then there's artificial transformation. And this is where you use a special science trick to make that happen. Naturally, bacteria will do this trick, not frequently, but it happens often enough every day. This is one way that bacterial cells can absorb new DNA from the environment, say from the walls of a hospital where bacterial cells had already been killed, all right? So here you have a cell, a bacterial cell, that has a receptor that allows a DNA fragment to be brought into the cell. Crazy, right? So here's the DNA fragment being brought into the cell. The DNA fragment can then become incorporated into the host genome by using a process that's really, really similar to crossing over. That little chunk of DNA ends up getting incorporated into the host chromosome, and now, boom, the host cell has new DNA to work with. So sometimes DNA enters in a controlled fashion, where you have what we call homologous recombination. It's like crossing over occur. Sometimes you have DNA incorporated into the new chromosome at a random site. That's non-homologous recombination, or they call it illegitimate recombination. We're not going to cover how that happens. Don't worry about that right now. But the idea is that you have bacteria preferentially taking up DNA from dead bacteria out in the environment. And it turns out that bacteria can take up chunks that are between 9 and 10 base pairs long, all the way up to hundreds of base pairs long, thousands even, all right? This is called horizontal gene transfer. Horizontal gene transfer is where you transfer genes from one organism to another without generating offspring. Vertical gene transfer is offspring, fertilization, offspring formation. Horizontal gene transfer is where you have transformation, transduction, or not cell mating, conjugation occurring. So vertical gene transfer always produces offspring. Horizontal gene transfer is going to be transforming cells. So it turns out 17% of E. coli and streptomycin genes have come about through incorporation of E. coli and streptococcus over the past 100 million years. So what does that do? That generates situations where you have organisms that can become, that cause rare diseases, organisms that become resistant to antibiotics. Again, that is a big deal in today's world. If you take antibiotics to kill a bacterium, you need to finish taking them all, because the dead bacteria living in your lungs or in your sinuses or in your skin are leaving fragments of DNA in those environments that living cells can absorb. Through horizontal gene transfer, we can have a huge generation of antibiotic-resistant organisms. That is not a good thing. And in fact, nearly 50% of Streptococcus pneumoniae throughout the world are penicillin resistant through this simple naturally occurring transformation in the environment. I don't mean to scare you, but please, if you take antibiotics, finish them up. Let your body heal. Complete the run, because we know that the amount of time that you're given antibiotics will wipe out enough cells such that we won't get resistant organisms. All right? Thank you guys for finishing up with Chapter 9. I'll see you in Chapter 10.